Welcome to the Deep Sleep Science channel. Tonight, we're gently exploring one of the most remarkable systems within you, the nervous system. If you enjoy calm, science-based journeys like this, consider liking or subscribing so you can return to quieter explorations whenever you need them. The nervous system is the body's communication network. It is made up of the brain, the spinal cord, and all the nerves that branch out to every organ, muscle, and patch of skin. It constantly gathers information from inside and outside your body, processes that information, and sends instructions back out often in fractions of a second. It allows you to think, feel, move, remember, digest food, regulate your heartbeat, and even breathe without conscious effort. Through tiny electrical signals and chemical messengers, billions of specialized cells coordinate your internal world with the world around you. The blood-brain barrier is a highly selective boundary that carefully manages what can move from the bloodstream into brain tissue. You can think of it as a protective filtering system built into the smallest blood vessels of the brain, helping keep the brain's surroundings stable. By limiting sudden chemical changes, it supports the precise conditions neurons need to signal reliably. This selectivity is one reason the brain can function with such consistency, even when the rest of the body is adapting to meals, exercise, stress, or temperature shifts. Tight junctions between brain microvascular endothelial cells are a central structural feature that restricts paracellular permeability at the blood-brain barrier. These junctions act like closely sealed seams between neighboring cells, so substances can't simply slip through the gaps. Instead, many molecules must use controlled, pathways-like specific transport proteins to cross. This structural sealing is one of the reasons brain capillaries behave differently from many other capillaries in the body. Astrocyte end feet wrap around tiny CNS blood vessels and provide biochemical support that helps maintain blood-brain barrier properties. Their close contact with the vessel walls allows them to influence how the barrier behaves and how well it stays regulated. Astrocytes can release signals that help endothelial cells maintain their specialized barrier identity. In this way, brain cells and blood vessels cooperate to keep the neural environment steady. Pericytes embedded in the capillary basement membrane are also part of the cellular architecture often described as supporting blood-brain barrier function. They sit alongside the smallest blood vessels and interact with endothelial cells and other nearby structures. Their presence is associated with stability and regulation of the microvascular wall. In many descriptions, they are considered an important partner in maintaining healthy barrier behavior. The blood-brain barrier limits entry of many large or hydrophilic molecules, contributing to the brain's specialized chemical environment. Many substances that move freely elsewhere in the body face strict limitations at this boundary. This helps prevent random fluctuations from disturbing sensitive neural circuits. It also means the brain's extracellular fluid can remain more controlled than the general bloodstream. Some small nonpolar molecules, such as oxygen and carbon dioxide, can diffuse across the blood-brain barrier more readily than large polar compounds. Because they are small and can pass through cell membranes more easily, they move across this interface without needing special carriers. That's important because the brain constantly needs oxygen, and it must continuously remove carbon dioxide. This kind of diffusion supports the basic chemistry required for ongoing neural activity. Specialized transporters at the blood-brain barrier support entry of key nutrients, such as glucose and certain amino acids, needed for neural function. Rather than letting everything pass, the barrier uses selective doorways for specific necessities. Glucose transport is especially important because neurons rely heavily on a steady energy supply. These transport systems help deliver what the brain needs while still limiting unwanted substances. Blood-brain barrier selectivity is clinically important because many drugs do not easily reach the brain in therapeutic concentrations. A medication can circulate through the body and still have difficulty entering brain tissue. This is one reason treatments for brain disorders can be challenging to design. Researchers often consider how a drug's size, polarity, and transport options affect whether it can cross effectively. Cerebrospinal fluid is a clear fluid that bathes the brain and spinal cord, circulating through ventricles and the subarachnoid space. It forms a protective surrounding layer that separates delicate neural tissue from direct contact with bone and other structures. Its circulation connects brain spaces with regions around the spinal cord, creating a continuous fluid environment. This fluid helps keep conditions more stable across the central nervous system. Most cerebrospinal fluid is produced by the choroid plexus located within the lateral third and fourth ventricles. The choroid plexus is a specialized tissue that sits inside these fluid-filled spaces and actively contributes to CSF formation. Rather than being a passive leak, CSF production involves cellular processes that move water and solutes in controlled ways. This is part of how the central nervous system maintains its own internal fluid environment. Medical references describe that a large fraction of cerebrospinal fluid production commonly cited, around about 80%, is associated with the choroid plexus. This highlights how central the choroid plexus is to the fluid system around the brain and spinal cord. Continuous production supports continuous circulation, which helps maintain composition and pressure within normal ranges. It also reflects how active the brain's protective fluid system really is. Cerebrospinal fluid helps cushion the central nervous system against mechanical forces, acting as a shock-absorbing buffer. When you move your head, walk, or shift posture, the brain and spinal cord are not meant to be jostled directly against rigid structures. CSF provides a protective layer that reduces mechanical stress. This cushioning helps preserve delicate tissue over a lifetime of everyday motion. Cerebrospinal fluid volume in humans is commonly described as around 125–150 ml at a given time, even though much more is produced each day. This means the body is constantly turning over and renewing this fluid, rather than keeping the same batch indefinitely. The standing volume is relatively modest, while production and absorption continue in the background. This steady cycling helps maintain a consistent environment for the brain and spinal cord. Some references describe roughly about 500 ml of cerebrospinal fluid produced per day in adults, emphasizing continuous turnover. Over a day, that amount is several times the typical volume present at any one moment. This highlights how dynamic the CSF system is, with ongoing production and reabsorption. Continuous turnover supports both stability and clearance functions. The enteric nervous system contains hundreds of millions of neurons, making it one of the most complex neural networks outside the brain. These neurons are spread throughout the digestive tract, forming circuits that can sense and respond locally. Because digestion requires constant adjustment, movement, secretion, blood flow, this large neural population helps manage those demands. It's a powerful reminder that nervous system extends far beyond the head and spine. Some descriptions estimate the enteric nervous system includes roughly 200–600 million neurons in humans. Even the lower end of that range represents an enormous number of nerve cells dedicated to digestion-related control. This scale helps explain why the gut can coordinate complex patterns of movement and secretion on its own. It also helps explain why gut function can feel so responsive to changes in the body's internal state. The vagus nerve is a major communication pathway between the gut and brain, carrying extensive sensory information. Many fibers traveling in this nerve are sensory, bringing internal status updates toward the brain. Signals can reflect stretching, chemical conditions, and other internal cues. This provides one major route for the body's internal environment to be represented within the nervous system. The gut-brain axis describes bidirectional signaling among the central nervous system, the enteric nervous system, and additional endocrine and immune pathways. It is not just a single nerve connection, but a broader network of communication routes. Hormones, immune signals, and neural pathways can all contribute to how signals travel back and forth. This is why gut physiology and brain physiology are often discussed together. Neurons are excitable cells specialized for rapid electrical signaling, while glial cells provide support and maintenance functions. Neurons are built to generate and transmit fast signals, often over long distances. Glial cells, in contrast, are essential for keeping the environment stable, providing protection and supporting ongoing function. Together, they form a cooperative system where fast signaling depends on steady support. Glial cells are considered the most abundant cell type in the central nervous system. This means that when you picture brain tissue, a large proportion of the cells are not neurons. These supportive cells help manage the conditions that allow neurons to operate properly. Their abundance highlights how much work goes into maintenance, protection, and regulation within neural tissue. Myelin is not continuous along an axon. It is interrupted by small gaps called nodes of Ranvier. These gaps are not errors or missing pieces. They are part of the design of rapid signaling. They create repeating segments along the axon where electrical activity can be refreshed. This pattern supports efficient long-distance communication. Nodes of Ranvier are key sites for rapid saltatory conduction, allowing impulses to effectively jump from node to node. Instead of an electrical signal traveling smoothly along every millimeter of axon membrane, it can leap in steps between these gaps. This makes transmission much faster and more efficient for many myelinated fibers. It's one of the mechanisms that allows rapid coordination across the body. The nervous system is commonly divided into the central nervous system, brain and spinal cord, and the peripheral nervous system, which includes nerves outside them. This division reflects both anatomy and function. The CNS is the main processing and integration center, while the PNS connects that center to the rest of the body. Together, they form a continuous communication network. Information flows inward from sensors and outward to muscles and organs. The peripheral nervous system includes sensory pathways that bring information to the CNS and motor pathways that send commands outward. Sensory fibers carry messages about touch, temperature, pain, and internal states toward the spinal cord and brain. Motor fibers carry signals from the CNS to muscles and glands to produce actions and responses. This constant two-way traffic helps the body adjust from moment to moment. The peripheral nervous system is often subdivided into the somatic nervous system and the autonomic nervous system. The somatic division is commonly associated with voluntary control of skeletal muscles and many conscious sensations. The autonomic division is more focused on involuntary regulation of organs and internal functions. This split helps describe how the body manages both deliberate actions and automatic maintenance. The autonomic nervous system has three divisions: sympathetic, parasympathetic, and enteric. Each division has distinct patterns of influence and organization. The sympathetic division is often associated with mobilizing resources, the parasympathetic with restorative functions, and the enteric with digestive control. Together, they regulate many involuntary processes without requiring conscious effort. The sympathetic nervous system supports rapid mobilization of energy and resources during stress responses. It can increase readiness by shifting physiology toward quick action. This can involve changes like boosting cardiovascular output and reallocating energy availability. The goal is to prepare the body to respond efficiently to demand. The parasympathetic nervous system generally promotes rest and digest functions that conserve energy and support digestion. It is associated with calming physiological states and supporting recovery. Many of its influences help return organ systems toward baseline conditions after stress. This supports long-term stability and maintenance. Sympathetic and parasympathetic influences can act in opposing ways on the same organs to help maintain physiological balance. An organ can receive inputs from both divisions, which allows fine control rather than a single fixed setting. The balance can shift with context, such as activity, rest, temperature, or emotional state. This coordinated opposition helps keep internal conditions within healthy ranges. The autonomic nervous system regulates involuntary processes such as heart rate, blood pressure, respiration, digestion, and sexual arousal. Many of these processes must be adjusted continuously, even when attention is elsewhere. Autonomic pathways help coordinate these functions across multiple organ systems. This keeps internal physiology responsive and stable. The enteric nervous system is embedded within the gastrointestinal tract and can coordinate many digestive functions. It is positioned right where digestion happens, allowing rapid local control of movement and secretion. It can coordinate patterns like peristalsis and other motility behaviors. This embedded placement supports quick responses to local conditions inside the gut. The enteric nervous system is organized in major plexuses, including the myenteric plexus and submucosal plexus. These plexuses form networks of neurons distributed through layers of the gut wall. Their arrangement supports both muscle control and regulation of local environment. This structure helps digestive function stay organized along the tract. The myenteric plexus is strongly involved in coordinating gastrointestinal motility and peristalsis. It lies in a position well suited to influence the smooth muscle layers that move food along. Coordinated contractions require timing and patterning, and this plexus helps manage that. It contributes to the wave-like motion that propels contents forward. The submucosal plexus is involved in local sensing and regulation of secretion and blood flow in the gut. It helps coordinate how the gut lining responds to what is present inside the digestive tract. This can include adjusting secretions that support digestion and controlling local circulation. These changes help match resources to digestive needs. The enteric nervous system can operate with substantial autonomy, even when communication with the CNS is reduced. Local circuits can generate and coordinate digestive patterns without needing constant instructions from the brain. This autonomy supports reliable digestion during sleep, distraction, or injury that reduces central input. It also shows how distributed control can be built into the body. The human body has 12 pairs of cranial nerves that emerge from the brain. These nerves connect the brain to structures in the head, neck, and beyond, depending on the nerve. They carry sensory information, motor commands, and autonomic signals. Each pair serves distinct roles that can be tested in a neurological exam. Cranial nerves support sensory functions like smell, vision, and hearing, and motor functions like eye and facial movements. Some cranial nerves specialize in a single sensory channel, while others coordinate multiple functions. Together, they support perception and the fine movements involved in speaking, swallowing, and facial expression. Their organization reflects how the nervous system links sensation to action. The human body has 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. These pairs emerge segment by segment from the spinal cord region. They distribute throughout the trunk and limbs, carrying signals both to and from the CNS. This segmental arrangement supports systematic mapping between spinal levels and body regions. Spinal nerves are mixed nerves, typically carrying both sensory and motor fibers. This means a single spinal nerve can include pathways bringing information inward and pathways carrying commands outward. Mixed composition allows efficient bundling of different signal types. It supports coordinated function in the areas each spinal nerve supplies. Each spinal nerve forms from the union of dorsal, sensory, and ventral motor roots. The dorsal root conveys sensory input toward the spinal cord, while the ventral root conveys motor output away from it. When they merge, the resulting spinal nerve carries both kinds of fibers. This structural layout supports the mixed nature of spinal nerves. The spinal cord is described as segmentally organized, with 31 segments corresponding to the exiting spinal nerve pairs. Each segment is associated with a set of roots that serve specific body regions. This organization helps explain patterns of reflexes, sensation, and muscle control. It also supports clinical localization when certain functions are affected. There are 8 pairs of cervical spinal nerves, even though there are only 7 cervical vertebrae, because of how the roots exit near the skull and upper spine. The numbering of nerves follows the pattern of how they emerge relative to vertebrae, which shifts at certain transition points. This is a good example of how anatomical naming reflects developmental and structural arrangements. It also helps clarify why SI8 exists, even without an 8th cervical vertebra. Dermatomes are skin areas primarily supplied by sensory fibers from specific spinal nerve roots. These areas form a map of how sensory input from the skin connects back to the spinal cord. Dermatomes can overlap, but they still provide helpful patterns. They're often used to interpret where a sensory change might be coming from. Clinicians use dermatome patterns to help localize nerve root or spinal cord problems based on sensory changes. If numbness, tingling, or altered sensation appears in a specific region, dermatome maps can offer clues about which spinal level may be involved. This helps narrow down possible sources of symptoms. It's a practical example of anatomy guiding diagnosis. Some descriptions note 31 pairs of spinal nerves, but about 30 dermatomes because the C1 nerve typically does not contribute to cutaneous sensation. In other words, not every spinal nerve pair corresponds neatly to a skin region with clear sensation mapping. This exception is commonly mentioned in dermatome discussions. It helps explain why the dermatome count can differ slightly from the nerve pair count. The first 2 cranial nerves, the olfactory and optic nerves, arise from the cerebrum, while many of the others emerge from the brainstem. This reflects how smell and vision are closely connected to higher brain structures involved in perception and interpretation. In contrast, many other cranial nerves connect to the brainstem, which plays a central role in coordinating movement, reflexes, and vital autonomic functions. This arrangement shows how different parts of the brain specialize in handling different kinds of information. Cranial nerve 1, the olfactory nerve, primarily mediates smell. Specialized receptor cells high inside the nasal cavity detect odor molecules and convert them into electrical signals. These signals travel directly to brain regions involved in processing scent and linking it to memory and emotion. Because of this direct pathway, smells can often evoke vivid recollections and strong feelings. Cranial nerve 2, the optic nerve, primarily mediates vision. Light striking the retina is transformed into electrical signals that travel along this nerve toward the brain. From there, visual information is processed into shapes, colors, movement, and depth. The optic nerve is essential for transforming simple light patterns into the rich visual world we experience. Cranial nerve 3, the oculomotor nerve, contributes to eye movements and pupil responses. It controls several muscles that move the eye in different directions, allowing coordinated tracking of objects. It also influences the size of the pupil in response to light. Through these actions, it helps stabilize and adjust vision in changing environments. Cranial nerve, the 4th, the trochlear nerve, innervates the superior oblique muscle and contributes to precise eye movement control. This small but important nerve helps rotate the eye and coordinate vertical and diagonal movements. Its function supports smooth visual tracking and alignment between both eyes. Even subtle eye movements depend on its accurate signaling. Cranial nerve 5, the trigeminal nerve, carries facial sensation and controls muscles of mastication. It transmits touch, pain, and temperature information from the face toward the brain. It also activates muscles used for chewing. Because it has both sensory and motor roles, it is considered a mixed cranial nerve. Cranial nerve V, the abducens nerve, controls the lateral rectus muscle for outward eye movement. This allows each eye to move away from the midline. Coordinated activity between the abducens nerve and other eye movement nerves ensures that both eyes remain aligned. This coordination is necessary for stable, single vision. Cranial nerve the 7th, the facial nerve, supports facial expression and carries taste from part of the tongue while influencing tear and salivary glands. It activates muscles that allow smiling, frowning, and other expressions. It also helps regulate secretions that keep the eyes and mouth moist. Its diverse functions illustrate how one nerve can influence movement, sensation, and gland activity. Cranial nerve the 8th, the vestibulocochlear nerve, supports hearing and balance. One branch carries sound information from the inner ear to the brain. The other branch transmits signals related to head position and movement, helping maintain equilibrium. Together, these functions allow awareness of sound and stable posture. Cranial nerve the 9th, the glossopharyngeal nerve, contributes to swallowing and carries taste and sensory information from the throat region. It helps coordinate muscles involved in swallowing. It also transmits sensations from parts of the tongue and pharynx. This coordination is essential for safe and effective swallowing. Cranial nerve X, the vagus nerve, is a far-reaching nerve with major roles in parasympathetic control of the heart, lungs, and digestive organs. It carries signals that influence heart rate, breathing patterns, and digestive activity. Many of its fibers are sensory, sending information back to the brain about internal organ states. Its broad distribution makes it central to autonomic regulation. Cranial nerve Xi, the accessory nerve, contributes to neck and shoulder muscle control, supporting head movement. It activates muscles that allow turning the head and elevating the shoulders. These movements are important for posture and orientation. Even simple motions like looking to the side rely on its function. Cranial nerve the 12th, the hypoglossal nerve, controls tongue movements important for speech and swallowing. It allows the tongue to move precisely in multiple directions. These movements shape words during speech and help position food during chewing and swallowing. Proper tongue coordination is vital for both communication and nutrition. The vestibulocochlear nerve carries distinct auditory and balance information via its cochlear and vestibular components. The cochlear part processes sound waves into neural signals. The vestibular part detects head motion and spatial orientation. Together, they help coordinate hearing with physical balance. Some cranial nerves are purely sensory, some purely motor, and some mixed, reflecting diverse roles. Sensory nerves specialize in bringing information inward. Motor nerves send commands outward, and mixed nerves handle both. This classification helps describe how each nerve contributes to overall function. It also provides a structured way to understand neurological examinations. The cranial nerves collectively support many head and neck functions, including sensory input, motor control, and autonomic regulation. They enable vision, hearing, facial expression, swallowing, and more. Because they connect directly to the brain, they provide efficient pathways for complex control. Their organization reflects how essential head and neck functions are to daily life. The peripheral nervous system includes cranial nerves and spinal nerves, linking the central nervous system to sensory receptors, muscles, and organs. It acts as the communication highway between the body and the brain and spinal cord. Signals from the skin, muscles, and organs travel inward, while commands travel outward. This network allows coordinated responses to both internal and external changes. Spinal nerves connect the spinal cord to specific regions of the body, distributing sensory and motor innervation segmentally. Each pair corresponds to a particular body level. This organized distribution allows predictable patterns of sensation and movement. It also explains why certain injuries affect specific areas. Spinal nerves carry information to and from the trunk and limbs and are central to reflexes and voluntary movement coordination. They participate in rapid reflex arcs that do not require conscious thought. They also relay voluntary commands from the brain to muscles. This dual role supports both automatic and intentional actions. The spinal cord is divided into cervical, thoracic, lumbar, sacral, and coccygeal regions matching spinal nerve groupings. Each region supplies different parts of the body. Cervical segments relate largely to the neck and upper limbs, thoracic to the trunk, and lumbar and sacral to the lower body. This structural organization mirrors functional distribution. The organization of spinal roots includes dorsal rootlets and ventral rootlets that fuse to form dorsal and ventral roots. Dorsal components carry sensory input toward the spinal cord. Ventral components carry motor output away from it. When they merge, they form the mixed spinal nerve. The first cervical spinal nerve root exits above the C1 vertebra, illustrating how nerve numbering and vertebral numbering can differ. This arrangement results from how nerves and bones develop and align. It shows that anatomical naming reflects structural relationships rather than simple counting. Understanding this helps interpret clinical imaging and anatomy. The C8 spinal nerve root exits between the C7 and T1 vertebrae, reflecting the extracervical nerve pair. This is why there are 8 cervical nerve pairs but only 7 cervical vertebrae. The numbering shifts after this point so that nerves exit below their corresponding vertebrae. It's a small but important detail in spinal anatomy. The spinal cord's segmental organization supports clinical mapping of symptoms to spinal levels via dermatomes and myotomes. Myotomes represent muscle groups supplied by specific spinal segments. Together with dermatomes, they help clinicians localize neurological issues. This mapping connects anatomy directly to function. The autonomic nervous system influences respiration, digestion, and cardiovascular parameters without requiring conscious effort. It adjusts heart rate, vessel diameter, and digestive activity continuously. These adjustments respond to internal and external cues. Much of this regulation happens outside awareness. Parasympathetic pathways are described as involving preganglionic and postganglionic neurons arranged in sequence. A signal leaves the central nervous system, synapses in a ganglion, and then continues to the target organ. This two-neuron chain allows distributed control. It also creates opportunities for modulation along the pathway. Sympathetic activation is associated with fight-or-flight physiology, while parasympathetic activation supports restorative processes. These two divisions are just organ systems in complementary ways. One prepares the body for action, the other encourages recovery and conservation. Together, they allow flexible adaptation. In the autonomic nervous system, signals often pass through peripheral ganglia before reaching target organs. These ganglia serve as relay stations. They help coordinate and distribute signals. This layered structure supports both precision and flexibility. Parasympathetic activity is commonly associated with slowing heart rate and promoting digestion. It encourages energy conservation and nutrient absorption. During restful states, parasympathetic influence is often more prominent. This supports long-term maintenance. Sympathetic activity is commonly associated with increasing cardiovascular output and mobilizing energy availability during acute stress. It helps redirect blood flow and increase readiness. This can involve changes in heart rate and metabolic activity. The goal is efficient response to demand. The autonomic nervous system's core role is maintaining internal homeostasis while adapting to changing demands and stresses. It constantly monitors and adjusts organ function. This balance supports stable internal conditions. Adaptation and stability work together. Sympathetic and parasympathetic outputs are not simply on or off, they vary by organ and context. Different organs may respond differently at the same time. This nuanced control allows fine-tuning rather than extremes. It reflects a dynamic regulatory system. The enteric nervous system coordinates digestive movements and influences secretion of gastrointestinal enzymes. It manages the mechanical and chemical aspects of digestion. Movement and secretion must be synchronized. Enteric circuits support that coordination. Enteric circuits interact with immune and endocrine signals within the gut environment. Hormones and immune mediators influence neural activity. Neural responses can also influence immune and endocrine processes. This creates a multi-system regulatory network. The gut-brain axis links emotional and cognitive centers to intestinal function through bidirectional communication pathways. Signals can travel from the brain to the gut and from the gut to the brain. This helps explain why emotional states can influence digestion. It also explains why gut states can affect mood and perception. The enteric nervous system includes sensory neurons that detect gut conditions and interneurons that integrate local reflexes. Sensory cells monitor stretch, chemicals, and other factors. Interneurons process this information. Together they generate coordinated responses. Enteric motor neurons coordinate smooth muscle contractions that move contents through the gastrointestinal tract. They create rhythmic patterns that propel food. These movements must be precisely timed. Neural control ensures organized progression. The nervous system uses both electrical signaling through action potentials and chemical signaling through synaptic transmission to communicate. Electrical signals travel quickly along axons. Chemical signals bridge the tiny gap between cells. Together they allow rapid yet flexible communication. The resting membrane potential depends on ion gradients and selective membrane permeability, especially for potassium. Neurons maintain differences in ion concentration inside and outside the cell. These differences create a voltage across the membrane. This resting state prepares the neuron for activation. Action potentials are initiated when depolarization reaches threshold, triggering a rapid and stereotyped response. Once threshold is reached, ion channels open in a coordinated way. This produces a predictable spike in voltage. The signal then travels along the axon. Voltage-gated channel timing first sodium, then potassium is fundamental to depolarization and repolarization. Sodium channels open quickly to create the rising phase. Potassium channels open slightly later to restore the membrane toward resting state. This sequence shapes the action potential. Myelination increases conduction speed by reducing current leak and concentrating regeneration at nodes of Ranvier. The insulating myelin sheath allows signals to travel efficiently. Instead of spreading continuously, impulses are refreshed at nodes. This supports rapid long-distance communication. Some axons are unmyelinated, and their conduction is generally slower than myelinated fibers of similar diameter. Without insulation, electrical signals spread more gradually. these fibers still serve important functions. Their slower speed suits certain types of signaling. The central nervous system and peripheral nervous system use shared molecular mechanisms such as ion channels and synapses, but differ in injury response and repair capacity. The underlying signaling machinery is similar. However, regeneration outcomes vary. This contrast is an important topic in neurobiology. The nervous system's organization allows rapid reflex-like responses and slower integrative responses, depending on pathway length and synaptic processing. Simple reflex arcs can operate quickly with minimal processing. More complex responses involve multiple synapses and brain regions. Both types of processing are essential. Many involuntary organ functions are regulated through autonomic pathways that do not require cortical involvement for moment-to-moment control. Heartbeat and digestion continue during sleep or distraction. Higher brain regions can influence them, but basic regulation does not depend on conscious thought. This ensures continuity of vital functions. Autonomic dysfunction can affect multiple systems simultaneously, because autonomic fibers innervate heart, vessels, lungs, gut, and glands. When regulation is disrupted, changes may appear in several organs at once. This reflects the wide distribution of autonomic pathways. It also shows how integrated the system is. Across the nervous system, coordinated electrical conduction, chemical neurotransmission, and glial maintenance together enable perception, movement, autonomic control, and internal stability. Neurons generate and transmit signals. Synapses translate those signals between cells. Glial cells maintain the environment that allows everything to function smoothly. Neurons are often categorized functionally as sensory neurons, motor neurons, and interneurons. Sensory neurons specialize in detecting changes in the environment or inside the body and carrying that information inward. Motor neurons transmit commands from the central nervous system to muscles or glands, allowing action to occur. Interneurons connect neurons within the brain and spinal cord, forming complex networks that process and integrate information. Sensory neurons transmit information from receptors toward the central nervous system. These receptors can detect touch, temperature, pressure, pain, light, sound, or chemical signals. Once activated, sensory neurons convert these stimuli into electrical impulses. Those impulses travel along nerve fibers toward the spinal cord and brain for interpretation. Motor neurons transmit commands from the central nervous system to muscles and glands. When the brain or spinal cord decides on an action, motor neurons carry that decision outward. In skeletal muscles, this can result in voluntary movement. In glands or smooth muscle, it can trigger secretion or involuntary adjustments. Interneurons form much of the central nervous system circuitry by connecting neurons within the brain and spinal cord. They often do not send signals directly to muscles or from sensory receptors. Instead, they act as processors and integrators of information. Through vast networks of interneurons, the nervous system can compare, evaluate, and coordinate responses. Neurons can be classified structurally as multipolar, bipolar, or pseudo-unipolar based on the number and arrangement of processes extending from the cell body. This classification reflects how many projections a neuron has and how they are organized. The shape of a neuron often relates to its function. Structural design supports efficient signal transmission in different contexts. Multipolar neurons typically have one axon and multiple dendrites, and are common among motor neurons and many interneurons. The multiple dendrites allow them to receive input from many other neurons. This makes them well suited for integration and coordination. Their structure supports complex processing. Bipolar neurons have one dendrite and one axon and are associated with certain special senses. They are commonly found in sensory pathways such as those for vision or smell. Their simpler structure supports more direct transmission of sensory information. This streamlined design fits specialized sensory roles. Pseudo-unipolar neurons have a single process that splits into two branches and are typical of many sensory neurons. One branch carries signals from the periphery toward the cell body, and the other continues toward the spinal cord. This arrangement allows rapid transmission of sensory information. It is especially common in spinal sensory pathways. The nervous system communicates chemically at synapses using neurotransmitters released from presynaptic terminals. When an electrical impulse reaches the end of an axon, it triggers release of chemical messengers. These chemicals cross a tiny gap between cells. They then bind to receptors on the next cell to continue the signal. Neurotransmitters are chemical messengers that carry signals from one neuron across a synaptic cleft to another neuron or to an effector cell. The synaptic cleft is an extremely small space between cells. Neurotransmitters diffuse across this gap and bind to specific receptor proteins. This binding changes the electrical or chemical state of the receiving cell. Neurotransmitters can be excitatory, inhibitory, or modulatory, depending on receptor type and cellular context. An excitatory neurotransmitter increases the likelihood that the next neuron will fire. An inhibitory neurotransmitter decreases that likelihood. A modulatory neurotransmitter can fine-tune or adjust how neurons respond over time. Glutamate is the principal excitatory neurotransmitter in much of the brain. It plays a central role in activating neural circuits. When glutamate binds to its receptors, it typically makes neurons more likely to generate action potentials. Because of its widespread use, it is fundamental to many brain processes. GABA is a major inhibitory neurotransmitter in the adult vertebrate brain. It reduces the likelihood that a neuron will fire an action potential. By providing inhibitory balance, GABA helps prevent excessive excitation. This balance is important for stable neural activity. Glycine is another inhibitory neurotransmitter that is especially prominent in the spinal cord. It plays a key role in shaping motor output and reflexes. By dampening certain signals, glycine helps coordinate muscle activity. Inhibition is just as important as excitation for smooth function. Acetylcholine is a major neurotransmitter in both the central and peripheral nervous systems. In the peripheral system, it is especially important at neuromuscular junctions. In the brain, it participates in attention and other processes. Its diverse roles reflect its importance. Dopamine, norepinephrine, and serotonin are examples of monoamine neurotransmitters with broad modulatory effects. Rather than simply turning neurons on or off, they adjust how circuits respond. They can influence mood, attention, motivation, and arousal. Their effects often extend across wide regions. Many neurotransmitters are cleared from synapses by reuptake into presynaptic terminals or surrounding cells, or by enzymatic degradation. This ensures that signals are brief and controlled. Without removal, neurotransmitters would continue to stimulate receptors. Clearance resets the synapse for the next signal. Synaptic transmission at the neuromuscular junction uses acetylcholine as the primary neurotransmitter. When a motor neuron fires, it releases acetylcholine into the synaptic cleft. This chemical binds to receptors on the muscle fiber. The result is muscle activation. The synaptic cleft at the neuromuscular junction is extremely narrow, often described as tens of nanometers wide. Despite its small size, it is critical for communication. The short distance allows rapid diffusion of acetylcholine. Precision in this tiny space supports reliable muscle control. At the neuromuscular junction, acetylcholine binds to nicotinic acetylcholine receptors on the muscle motor end plate to trigger depolarization. These receptors are ion channels that open when acetylcholine binds. Opening allows ions to flow, changing the muscle membrane potential. This initiates muscle contraction. Acetylcholinesterase in the synaptic cleft rapidly breaks down acetylcholine, limiting the duration of muscle activation. This enzyme ensures that the signal is brief. Once acetylcholine is broken down, receptors close. The muscle fiber can then relax and prepare for the next signal. Acetylcholinesterase is exceptionally efficient and can hydrolyze large numbers of acetylcholine molecules per unit time. This high efficiency ensures rapid signal termination. Without it, muscle activation could become prolonged and uncontrolled. Enzymatic precision supports smooth movement. Neuromuscular junctions include specialized postjunctional folds that increase surface area for receptors. These folds allow more receptors to be packed into a small area. Higher receptor density increases the reliability of transmission. Structural design enhances functional precision. Some toxins cause paralysis by interfering with acetylcholine release or breakdown at the neuromuscular junction. By disrupting transmitter release or preventing its degradation, these toxins alter normal signaling. This demonstrates how delicate synaptic balance is. Even small disruptions can have major effects. The neuromuscular junction uses quantal release of acetylcholine packets from synaptic vesicles. Each vesicle contains a set amount of neurotransmitter. When vesicles fuse with the membrane, they release their contents in discrete amounts. This quantal release allows precise control of signaling strength. The neuromuscular junction signaling depends on vesicle fusion mechanisms, including proteins like SNARE that control transmitter release. These proteins help vesicles merge with the presynaptic membrane. Without proper fusion, acetylcholine cannot enter the synaptic cleft. Molecular machinery enables each transmission event. Botulinum toxin can cause flaccid paralysis by inhibiting acetylcholine release at the neuromuscular junction. It interferes with the proteins required for vesicle fusion. As a result, acetylcholine cannot be released. Muscles then fail to receive activation signals. Disrupting acetylcholinesterase activity can cause excessive acetylcholine in the synaptic cleft, altering muscle function. If acetylcholine is not broken down efficiently, receptors remain activated longer than normal. This can lead to abnormal muscle contraction. Balanced breakdown is essential. Neurotransmitter receptors are proteins that convert chemical binding events into cellular responses, including ion fluxes or second messenger cascades. When a neurotransmitter binds, the receptor changes shape. This change can open ion channels or activate intracellular signaling pathways. Receptors translate chemical signals into electrical or biochemical changes. Synaptic signaling depends on neurotransmitter binding to receptors, which can be ionotropic or metabotropic. Ionotropic receptors act quickly by directly controlling ion channels. Metabotropic receptors act more slowly through second messenger systems. These two types allow both rapid and prolonged effects. The all-or-nothing property of action potentials means their amplitude is relatively constant once initiated. If the threshold is reached, the full spike occurs. If not, no spike occurs. This ensures consistent signal strength along the axon. An action potential is an all-or-nothing change in membrane voltage driven by ion channel dynamics. Voltage-gated sodium and potassium channels open and close in sequence. These changes produce a rapid rise and fall in voltage. The result is a standardized electrical pulse. The rising phase of a neuronal action potential is initiated by opening of voltage-gated sodium channels and sodium influx. Sodium ions move into the cell due to concentration and electrical gradients. This causes rapid depolarization. The membrane potential shifts toward positive values. Repolarization is primarily supported by sodium channel inactivation and opening of voltage-gated potassium channels. As sodium channels close, potassium channels allow potassium ions to exit. This moves the membrane potential back toward resting levels. The coordinated timing restores stability. Many neurons experience an after-hyperpolarization due to continued potassium conductance after repolarization. Potassium channels may remain open briefly. This causes the membrane potential to dip below resting level. This phase influences how soon another action potential can occur. The absolute refractory period largely reflects sodium channel inactivation, preventing immediate refiring. During this time, another action potential cannot be generated. This ensures that signals move in one direction. It also limits maximum firing frequency. The relative refractory period follows, when stronger stimulation may be required to trigger another action potential. During this time, the membrane is still returning to resting state. It is possible but more difficult to fire again. This shapes signal timing. Refractory periods help ensure action potentials propagate in one direction along an axon under normal conditions. Because recently activated regions cannot fire immediately again, signals move forward. This supports orderly transmission. Directional flow is crucial for coherent communication. Nodes of Ranvier are highly enriched in voltage-gated sodium channels, supporting rapid action potential regeneration. At these nodes, the electrical signal is boosted. High channel density ensures efficient depolarization. This is key to saltatory conduction. Myelinated axons are organized into internodes separated by nodes, forming repeating conduction units. Each internode is insulated by myelin. Each node allows signal regeneration. This repeating pattern supports speed and efficiency. Nodes of Ranvier can be extremely short relative to internodes, yet are functionally critical. Even though they occupy small segments, they house essential ion channels. Without them, myelinated conduction would not work. Small structures can have large effects. Myelin is formed by different glial cells depending on location. Oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. Although both create insulating sheaths, they differ in structure and behavior. This distinction influences repair capacity. Oligodendrocytes can extend multiple processes, allowing a single cell to myelinate segments of multiple axons in the central nervous system. This efficient design supports dense packing of axons. It also reflects differences from peripheral myelination. Schwann cells typically myelinate a single axon segment in the peripheral nervous system. Each Schwann cell wraps around one section of one axon. This one-to-one relationship supports distinct repair processes in peripheral nerves. Myelin-related differences between central and peripheral glia contribute to different repair outcomes after injury. Peripheral nerves often regenerate more effectively. Central nervous system injuries show more limited regrowth. Glial behavior plays a role in this contrast. After peripheral nerve injury, Wallerian degeneration occurs in the distal axon segment, and debris is cleared as part of repair. The damaged distal portion breaks down. Cells then remove debris. This prepares the pathway for potential regrowth. Schwann cells shift to a repair-supportive phenotype after axonal injury, aiding regrowth and guidance of regenerating axons. They change gene expression and behavior. They help guide new axonal sprouts. This supports recovery. Peripheral axons are often described as regenerating slowly, at rates on the order of about 1 to 2 millimeters per day under favorable conditions. This gradual process can take weeks or months. Distance to target influences recovery time. Regeneration requires coordinated cellular activity. Glial cells do not usually generate and propagate action potentials the way neurons do, but they still have a powerful influence on how neural circuits behave. Instead of acting like wires that carry fast electrical pulses, many glial cells act more like caretakers and regulators of the neural environment. They help neurons stay healthy, keep signaling stable, and adjust how strongly or efficiently networks communicate. In many modern views of neuroscience, glia are not passive helpers, they are active partners in brain function. Major central nervous system glial cell types include astrocytes, oligodendrocytes, microglia, and ependymal cells. Each type has its own role, and together they support nearly every aspect of neural life. Astrocytes help manage the chemical surroundings of neurons. Oligodendrocytes create insulating myelin in the CNS. Microglia handle immune-like surveillance and cleanup. And ependymal cells line fluid spaces. This diversity shows that glia is not one thing, but a family of specialized support systems. Major peripheral nervous system glial cell types include Schwann cells and satellite cells. Schwann cells are well known for their relationship to peripheral axons and for their role in myelination and repair. Satellite cells surround neuron cell bodies in peripheral ganglia and help regulate the local environment around those neurons. Together, these cells help peripheral nerves and ganglia remain stable and functional in a wide range of body conditions. The blood-brain barrier and the blood-cerebrospinal fluid barrier differ in permeability, and regions like the choroid plexus have more permeable capillaries than typical blood-brain barrier capillaries. This means the body uses different filtering styles depending on where the blood is interacting with the central nervous system. In brain tissue itself, the barrier is extremely tight, keeping many substances out. In the choroid plexus, the capillaries allow easier exchange, but the epithelial layer still tightly controls what enters the CSF. Cerebrospinal fluid circulation commonly proceeds from the lateral ventricles to the third ventricle via the interventricular foramina, then through the cerebral aqueduct to the fourth ventricle. This pathway is like a connected set of chambers and corridors inside the brain. The ventricles are fluid spaces, and their connections allow CSF to move through the system in an organized way. Understanding this route helps explain how blockage at a narrow point like the cerebral aqueduct can affect fluid flow and pressure. From the fourth ventricle, cerebrospinal fluid enters the subarachnoid space through openings that include a median aperture and paired lateral apertures. These openings provide a way for CSF to leave the internal ventricle system and flow into the spaces around the brain and spinal cord. Once in the subarachnoid space, the fluid can circulate widely and cushion neural tissue. This transition is important because it links internal fluid chambers to the protective outer fluid layer. Cerebrospinal fluid is absorbed back toward venous drainage pathways via arachnoid granulations, or villi in many traditional descriptions. These structures act like one-way valves that allow CSF to return toward the bloodstream while limiting backflow. The result is a continuous cycle: production, circulation, and reabsorption. This circulation helps maintain steady pressure and composition in the central nervous system's fluid spaces. Some research discussions emphasize that details of cerebrospinal fluid drainage routes can be more complex and have been actively revised by modern studies. In other words, scientists have been refining the story of exactly where CSF goes and how it leaves the cranial and spinal spaces. New evidence has highlighted additional pathways beyond the classic descriptions. This is an example of how anatomy is not only something to memorize, but also a subject where understanding can evolve as measurement tools improve. Cerebrospinal fluid production involves active transport processes across choroid plexus epithelial cells, not just passive filtration. The choroid plexus does not simply leak fluid out of blood vessels. Instead, cells move ions and other solutes in controlled ways, and water follows these gradients. This active control helps determine CSF composition and supports a stable environment for the brain and spinal cord. Cerebrospinal fluid has immunological roles, contributing to central nervous system protection in addition to mechanical cushioning. The brain and spinal cord are protected not only by bones and fluid, but also by immune-related surveillance and signaling. CSF can carry immune molecules and can reflect inflammatory changes. This is one reason CSF sampling can reveal signs of infection or immune activity in the CNS. Cerebrospinal fluid circulates through the central canal of the spinal cord, as well as around the brain and spinal cord in subarachnoid spaces. This means CSF is not limited to the skull, it extends down through the spinal system too. The continuity between cranial and spinal CSF spaces helps distribute cushioning and maintain consistent chemical conditions. It also helps explain why certain pressure changes can affect both head and spine-related symptoms. Cerebrospinal fluid secretion is described as having circadian variation in some discussions, though mechanisms remain under study. This suggests that CSF dynamics may not be identical at all hours of the day. The body often shows daily rhythms in hormones, temperature, and metabolism, and CSF production may also vary with time and physiological state. Researchers continue to investigate exactly how sleep-wake cycles and brain activity relate to CSF flow and turnover. Ependymal cells line the ventricles and are associated with cerebrospinal fluid-related structures, such as the choroid plexus epithelium. Their location places them at the boundary between neural tissue and fluid spaces. By lining the ventricles, they help define the surfaces where CSF moves. Their relationship with CSF spaces highlights how the nervous system includes not only neurons and synapses, but also specialized barriers and linings that support fluid dynamics. Many glial cells retain the ability to divide, unlike most mature neurons. This matters because dividing cells can respond differently to injury and change over time. In the adult nervous system, most neurons are considered long-lived and generally do not divide, while many glial cells can proliferate under certain conditions. This difference influences both repair processes and how the nervous system responds to damage or disease. The central nervous system generally shows less robust axonal regrowth after injury compared with the peripheral nervous system. When peripheral nerves are injured, regrowth and reconnection are more likely, although still slow and imperfect. In the CNS, regrowth is more limited, and functional recovery can be harder to achieve. This contrast is a major topic in neuroscience because it drives research into therapies and rehabilitation strategies. In the central nervous system, glial scarring can inhibit axon regrowth after injury. After damage, certain glial responses can form a scar-like environment that stabilizes the injury site, but also creates barriers for regrowing axons. This is a complicated trade-off. Protecting tissue can come at the cost of preventing reconnection. Understanding how and why glial scarring forms is part of the effort to improve CNS recovery. In the peripheral nervous system, Schwann cells are often described as promoting repair and remyelination after injury. Schwann cells can reorganize after damage to help clear debris, guide regrowing axons, and later restore insulation around repaired fibers. This supported behavior is one reason peripheral nerves tend to recover better than central pathways. Their activity shows how specific cell types can shape healing outcomes. Schwann cells can guide regenerating axons along basal lamina pathways in peripheral nerves. The basal lamina can act like a supportive corridor that helps regrowing axons find their way. Schwann cells help maintain and organize this pathway, increasing the chance that axons reconnect with appropriate targets. Guided regrowth is important because random growth would not restore precise function. In peripheral nerves, Schwann cells contribute to clearing degenerating debris and coordinating remyelination during regeneration. After injury, old myelin and damaged axon parts must be removed before effective repair can proceed. Schwann cells help manage this cleanup and then shift toward rebuilding insulation. This combination of cleanup and rebuilding helps explain why Schwann cells are central to peripheral nerve recovery. Schwann cell-based therapies are actively researched as potential tools to support peripheral nerve repair. Researchers explore ways to enhance the natural repair abilities of Schwann cells, or to use them in engineered approaches. This can include guiding regeneration, improving remyelination, or supporting reinnervation of muscles and skin. Such therapies aim to make recovery faster, more complete, or more reliable. Peripheral nerve regeneration research explores not only natural Schwann cell responses, but also transplantation and engineered strategies to improve outcomes. In addition to relying on the body's own repair mechanisms, scientists test grafts, scaffolds, and cell-based approaches. These strategies can create better pathways for axons to regrow or improve the environment around an injury. This field shows how biology and engineering often combine in modern medicine. Peripheral nerves can contain bundles of axons serving different targets, and injury location affects which functions are disrupted. A single nerve may include fibers for sensation, movement, and autonomic control. If an injury damages only part of a nerve bundle, symptoms can be selective, affecting certain sensations or muscles more than others. This bundled organization helps explain why nerve injuries can produce complex and sometimes patchy symptom patterns. Peripheral nerve recovery can vary widely depending on injury severity, distance to target, and whether guidance structures remain intact. A small injury with preserved pathways can allow more direct regrowth and better outcomes. A severe injury with disrupted structure makes it harder for axons to find their original targets. Distance matters too, because slow regrowth means long waits for faraway muscles or skin regions to be reinnervated. Regenerating axons may require timely reinnervation of muscle targets because prolonged denervation can reduce functional recovery. Muscles and other target tissues change when they are not receiving normal nerve input. Over time, they can weaken and lose some of their ability to respond, even if nerve fibers eventually return. This is why timing can influence how much function is regained after nerve damage. The neuromuscular junction contains acetylcholinesterase, anchored within the synaptic cleft region, helping confine transmitter action. By being positioned right where acetylcholine is released, the enzyme can break down the transmitter quickly and locally. This ensures that activation stays precise, rather than spreading or lingering too long. The placement of the enzyme is a structural strategy for controlling timing. The cholinergic system is clinically important because interfering with acetylcholine breakdown can dramatically alter muscle activation. If acetylcholine is not removed efficiently, muscles may contract too long or too strongly. If signaling is blocked, muscles may not activate when needed. This sensitivity is why acetylcholine-related pathways are central to understanding certain toxins and medications. Clearing acetylcholine rapidly is essential because prolonged activation at the neuromuscular junction would impair controlled movement. Smooth movement depends on rapid on-off timing, not continuous activation. If muscle fibers were held in a constant activated state, precise coordination would be lost. Rapid breakdown supports fine motor control and stable posture. The neuromuscular junction's reliability is supported by high receptor density and specialized synaptic architecture. The muscle side of the junction is designed to respond strongly to each nerve signal. Many receptors and structural folds help amplify the effect of neurotransmitter release. This makes transmission robust so that a motor neuron firing reliably produces muscle activation. Neurotransmitters are not limited to the brain. They also mediate signaling at peripheral synapses, including those affecting glands and smooth muscle. The same basic chemical communication style is used in many parts of the body. For example, autonomic nerves use neurotransmitters to influence heart muscle, blood vessels, and digestive tract movement. This shows that neurotransmission is a general principle of nervous system control, not just a brain-only feature. Neurotransmitter diversity is high, and some sources describe more than 40 neurotransmitters in the human nervous system. This diversity allows many distinct signaling styles and effects. Different transmitters can produce different time courses, strengths, and modulatory patterns. It also means that nervous system function is built from a complex chemical toolkit, not a single universal messenger. Neurotransmitter dysfunction can be linked to neurological and psychiatric conditions, reflecting the importance of synaptic chemistry and health. When transmitter systems are too strong, too weak, or poorly regulated, circuits can become unstable or unbalanced. This can influence mood, movement, cognition, and sensory processing. While conditions are complex and multifactorial, neurotransmitter systems remain a central focus in many explanations and treatments. Medications can act by altering neurotransmitter release, blocking receptors, inhibiting breakdown enzymes, or changing reuptake processes. These are different ways of adjusting the same signaling system. Some drugs increase availability of a neurotransmitter by preventing its removal, while others decrease signaling by blocking receptors. This variety shows how many control points exist in synaptic communication. Neurotransmitters are stored in synaptic vesicles and released in response to electrical activity at the presynaptic terminal. Vesicles keep neurotransmitters packaged and ready, preventing them from diffusing randomly inside the cell. When an action potential arrives, vesicles fuse and release transmitter into the synaptic cleft. This makes signaling fast, timed, and repeatable. After neurotransmitter release, removal mechanisms such as reuptake, enzymatic degradation, and diffusion terminate signaling and reset synapses. Without these cleanup processes, signals would blur together and timing would become unreliable. Clearing a synapse allows it to respond cleanly to the next impulse. In many systems, reuptake also recycles transmitter molecules or components for future use. Neurotransmitters can act on different receptors to produce different effects. The same transmitter may be excitatory in one tissue and inhibitory in another, depending on receptor subtype. What matters is not only the chemical itself, but the receptor that receives it. Different receptors open different channels or activate different intracellular pathways. This receptor-based logic gives the nervous system flexibility using a shared set of signaling molecules. Glutamate and GABA form a major excitatory-inhibitory balance in many central nervous system circuits, shaping stability and responsiveness. Excitation allows activity to spread and carry information forward, while inhibition prevents runaway firing and sharpens patterns. Balanced interaction helps networks remain stable but responsive. Many brain functions depend on this careful balance rather than extreme excitation or extreme inhibition. Glycine-mediated inhibition is especially significant in spinal cord circuits, including those that shape motor output. The spinal cord must coordinate timing across many muscles to produce smooth movement. Inhibitory signals help prevent conflicting muscle activation and help organize reflexes. Glycine's role in spinal inhibition demonstrates how chemical signaling supports motor coordination. Shingles, caused by varicella zoster reactivation, often follows a dermatomal distribution, producing a characteristic band-like pattern. This happens because the virus can lie dormant in sensory nerve ganglia and later reactivate. When it does, symptoms often appear along the sensory territory supplied by that nerve root. Dermatomes help explain why the rash and pain may appear in a stripe-like area rather than spreading randomly. Spinal nerve problems can arise from disc herniation or foraminal narrowing, potentially causing pain, weakness, or sensory changes. If a nerve root is compressed as it exits the spine, signals can be disrupted. Sensory fibers may produce numbness, tingling, or pain, while motor fibers may lead to weakness in specific muscles. The pattern of symptoms often matches the organization of spinal nerves and roots. Spinal nerves transmit autonomic impulses as well as sensory and motor signals, reflecting their mixed fiber content. Along with carrying touch and movement-related signals, spinal nerves can include fibers that influence sweating, blood vessel tone, and other involuntary functions. This is why some nerve injuries can affect skin temperature or sweating patterns, as well as sensation and movement. A single peripheral nerve can be a bundle of multiple functional systems. Many peripheral nerves include autonomic fibers that influence blood vessels, sweat glands, and other involuntary targets in addition to sensory and motor fibers. Autonomic fibers help regulate local blood flow and sweating, which support temperature control and skin function. Because these fibers travel alongside others, damage can create mixed symptoms. This bundled design is efficient for distributing multiple control signals to the same region. Dermatome mapping can help distinguish peripheral nerve injury from nerve root compression because the sensory patterns differ. A single peripheral nerve supplies a different pattern than a spinal root does. If numbness follows a dermatomal-like stripe, a root issue may be suspected if it follows a nerve distribution in the limb. A peripheral nerve problem may be more likely. This mapping helps clinicians reason from a symptom pattern back to anatomy. Dermatome patterns reflect spinal nerve root organization and can help clinicians interpret radiating pain or numbness. When pain travels down an arm or leg in a predictable route, dermatomes offer a framework for understanding why. This is not perfect, there is overlap, but it is often helpful. It connects the sensory map of the skin to the segmental structure of the spinal cord. The sympathetic nervous system can be activated by perceived threats, with brain circuits signaling downstream autonomic pathways. The body does not need to be physically harmed to prepare for action. Perception and interpretation can trigger autonomic shifts. Brain regions that evaluate threat can influence brainstem and spinal autonomic centers. This shows how cognition and physiology are connected through neural pathways. Parasympathetic function is discussed as supporting recovery from stress by helping return organ systems toward baseline. After a stress response, the body benefits from shifting toward calmer physiological settings. Parasympathetic influence can support digestion, slow heart rate, and encourage restorative processes. This recovery phase is part of maintaining long-term balance rather than staying in a constant alert mode. Autonomic balance involves coordinated regulation rather than constant maximal activation of either sympathetic or parasympathetic division. The body is not meant to be stuck fully in one extreme state. Instead, the two divisions adjust in shifting proportions depending on context activity, rest, temperature, and internal needs. This coordination supports stability across a wide range of conditions. The vagus nerve is notable among cranial nerves for its extensive distribution to thoracic and abdominal organs. It reaches beyond the head and neck, influencing heart, lungs, and much of the digestive system. This broad reach is why it is frequently mentioned in discussions of internal regulation. It acts as a major pathway for parasympathetic signals and for sensory information returning from organs. The vagus nerve transmits sensory state information from the gut toward the brain via vagal afferent fibers. Many vagal fibers carry information inward, reporting on stretch, chemical environment, and other internal cues. This helps the brain stay informed about digestive conditions without conscious awareness. It is part of the broader system that links internal physiology to brain regulation. The vagus nerve's broad organ influence helps explain why vagal signaling is frequently discussed in gut-brain communication. Because it connects to many digestive and thoracic organs, it can carry both control signals and sensory feedback. That combination supports bidirectional communication between brain and body. It also helps explain why researchers focus on vagal pathways when studying internal state regulation. The enteric nervous system is influenced by gut microbiota, which is one reason microbiome research intersects with neuroscience. Microbes can affect the gut environment and produce signals that influence immune and endocrine pathways. These changes can, in turn, influence enteric neural activity and gut function. This connection is one reason the gut is studied as part of broader nervous system-related health. Scientific reviews describe gut microbiota as influencing gut-brain signaling through immune, neuroendocrine, and vagal routes. This means the microbiome may affect signaling by changing immune activity, altering hormone-related signals, and influencing nerve pathways like the vagus. It is not a single direct line, but a network of routes. Understanding these routes helps explain why gut biology can be discussed in relation to brain-related outcomes. Gut-brain research links microbiota, immune signaling, and vagal pathways as contributors to how gut states can influence brain-related outcomes. The idea is that changes in the gut can shift signals traveling toward the brain through multiple channels at once. These channels can interact and amplify or dampen overall effects. This is why the gut-brain relationship is often described as complex and multi-layered, rather than simple cause and effect. The gut-brain axis integrates neural, immune, and endocrine signaling rather than relying on a single gut nerve. Neural pathways provide fast electrical routes, immune pathways provide chemical signaling linked to defense and inflammation, and endocrine pathways provide hormone-based messaging. Together, they create a coordinated system for communication. This integrated view explains why changes in one system can influence the others. The enteric nervous system is sometimes referred to as a second brain because of its complexity and partial autonomy. This nickname highlights that the gut contains large, organized neural networks capable of local processing. The enteric system can coordinate digestion without needing constant direction from the brain. While it is not a second brain in the full sense, the term reflects its remarkable independence and scale. Some descriptions note more than 100 million neurons lining the human digestive tract, emphasizing the scale of enteric circuitry. Even if exact counts vary by method and source, the key point is that the gut contains a very large nervous system. This supports complex patterns of movement, secretion, and sensing along the digestive tract. It also highlights why gut function can be so finely regulated. Enteric neurons are arranged in ganglia and plexuses along the length of the gastrointestinal tract. Ganglia are clusters of neuron cell bodies, and plexuses are networks of nerve fibers and ganglia. This organization creates repeating control regions along the digestive system. It helps coordinate local reflexes and larger-scale patterns like peristalsis. Some medical descriptions cite the enteric nervous system as operating within two thin layers of nerves, lining the gastrointestinal tract. These layers correspond to the major plexus regions embedded within the gut wall. Their placement allows them to interact directly with muscle layers and with the inner lining where sensing and secretion occur. This layered arrangement is a structural solution for controlling a long, dynamic tube. The gastrointestinal tract is densely innervated by enteric neurons and is also influenced by endocrine and immune signaling in its microenvironment. The gut is not just a digestive organ, it is an interactive biological environment. Local hormones influence digestion timing, immune cells respond to microbes and injury, and neurons coordinate movement and secretion. These systems overlap in the same space, allowing integrated control. The enteric nervous system regulates movements, secretions, blood flow, and reflexes like vomiting or diarrhea as part of gut defense and digestion control. Digestion requires the gut to move food, add enzymes and fluids, and adjust circulation to support absorption. Defensive reflexes can also be triggered to remove harmful substances. The enteric system helps coordinate these responses quickly and locally. Gut infections or inflammatory disruptions can change gut function partly through enteric neural responses affecting secretion and motility. When the gut detects irritation or infection, it may alter movement patterns and increase secretions. These changes can help flush or limit harmful factors. Enteric neurons contribute to coordinating these protective adjustments. The enteric nervous system contains hundreds of millions of neurons, making it one of the most complex neural networks outside the brain. This immense number allows it to coordinate detailed patterns of muscle contraction, secretion, and blood flow along the digestive tract. Rather than depending on constant instructions from the brain, it can process information locally and adjust digestive activity in real time. Its scale is one reason digestion is so adaptable and finely tuned. Some descriptions estimate the enteric nervous system includes roughly 200 to 600 million neurons in humans. While exact counts vary depending on measurement methods, the overall message is clear. The gut's neural network is extensive. That size supports a wide range of reflexes and coordination patterns. It also reinforces why the gut is sometimes described as having brain-like organizational features. The enteric nervous system is organized into major plexuses, including the myenteric plexus and the submucosal plexus. These plexuses are networks of interconnected neurons embedded within layers of the gut wall. Each plexus has a somewhat different functional emphasis. Together they create layered control over digestive movement and secretion. The myenteric plexus is strongly involved in coordinating gastrointestinal motility and peristalsis. Peristalsis is the wave-like contraction that moves food along the digestive tract. The myenteric plexus helps time and sequence these contractions so that material progresses smoothly. Without coordinated signaling, movement would be inefficient or disorganized. The submucosal plexus is involved in local sensing and regulation of secretion and blood flow in the gut. It helps adjust how much fluid and enzyme are released into the digestive tract. It also influences blood vessel dilation to match absorption needs. This local regulation supports effective digestion and nutrient uptake. The enteric nervous system can operate with substantial autonomy, even when communication with the central nervous system is reduced. Experiments and clinical observations show that many digestive patterns continue without direct brain input. Local circuits within the gut detect stretch, chemical composition, and other signals. They then generate appropriate reflex responses independently. Functional classifications of enteric neurons include motor neurons, intrinsic primary afferent neurons, interneurons, and intestinofugal neurons. Motor neurons influence smooth muscle and glands. Intrinsic primary afferent neurons detect local changes such as stretch or chemical composition. Interneurons connect and integrate signals within the plexus, and intestinofugal neurons send information outward to other ganglia. Enteric motor neurons coordinate smooth muscle contractions that move contents through the gastrointestinal tract. They determine when muscles contract and when they relax. This precise timing allows food to be mixed, broken down, and advanced along the tract. Coordinated contraction is essential for efficient digestion. The gut-brain axis describes bidirectional signaling among the central nervous system, the enteric nervous system, and additional endocrine and immune pathways. Signals do not flow in only one direction. The brain can influence digestion, and gut activity can influence brain function. This constant exchange helps maintain overall balance. The gut-brain axis is described as bidirectional, meaning signals travel both from brain to gut and from gut to brain. Brain states such as stress can change gut motility and secretion. At the same time, signals from the gut can affect mood, perception, and overall physiological state. This reciprocity is central to understanding whole-body regulation. The human body has 12 pairs of cranial nerves that emerge from the brain. These nerves connect the brain directly to structures in the head, neck, and in some cases, thoracic and abdominal organs. Each pair is specialized for certain sensory or motor functions. Their numbering from I to the 12th reflects their anatomical order. Cranial nerves support sensory functions like smell, vision, and hearing, and motor functions like eye and facial movements. Some are purely sensory, some purely motor, and others mixed. This diversity allows the brain to gather detailed information from the environment and coordinate precise responses. Cranial nerves are essential for everyday activities like speaking, chewing, and blinking. Cranial nerve 1, the olfactory nerve, primarily mediates smell. It carries signals from receptors in the nasal cavity to the brain. These signals allow detection of airborne chemicals. Smell contributes not only to sensory experience, but also to taste perception and environmental awareness. Cranial nerve 2, the optic nerve, primarily mediates vision. It transmits information from the retina to the brain. The visual signal then undergoes complex processing to interpret shapes, color, and movement. This nerve is essential for perceiving the external world. Cranial nerve 3, the oculomotor nerve, contributes to eye movements and pupil responses. It controls several muscles that move the eye in different directions. It also helps regulate pupil size and focus. Coordinated eye movement depends heavily on its function. Cranial nerve 4, the trochlear nerve, innervates the superior oblique muscle, contributing to eye movement control. Although it is small, its function is specific and important. It allows the eye to move downward and inward in a controlled way. Fine eye coordination requires precise input from multiple cranial nerves. Cranial nerve 5, the trigeminal nerve, carries facial sensation and controls muscles of mastication. It detects touch, pain, and temperature from much of the face. It also powers chewing muscles. This dual role highlights how sensory and motor functions can be combined in a single nerve. Cranial nerve V, the abducens nerve, controls the lateral rectus muscle for outward eye movement. This muscle allows the eye to move laterally. Proper function ensures both eyes can move together smoothly. Coordination between cranial nerves the 3rd, the 4th, and V supports stable vision. Cranial nerve the 7th, the facial nerve, supports facial expression and carries taste from part of the tongue while influencing tear and salivary glands. It enables smiling, frowning, and other facial movements. It also contributes to tear production and saliva secretion. This broad role makes it essential for communication and daily comfort. Cranial nerve the 8th, the vestibulocochlear nerve, supports hearing and balance. It has two main components: the cochlear branch for sound and the vestibular branch for balance and spatial orientation. Together they help maintain equilibrium and auditory perception. Damage can lead to dizziness or hearing loss, Cranial nerve the ninth, the glossopharyngeal nerve, contributes to swallowing and carries taste and sensory information from the throat region. It plays a role in coordinating swallowing reflexes. It also contributes to monitoring certain internal signals. Its function bridges sensation and motor coordination. Cranial nerve X, the vagus nerve, is a far-reaching nerve, with major roles in parasympathetic control of heart, lungs, and digestive organs. It extends beyond the head and neck into thoracic and abdominal regions. It influences heart rate, respiratory patterns, and digestive activity. Its widespread influence reflects its central importance. Cranial nerve C, the accessory nerve, contributes to neck and shoulder muscle control, supporting head movement. It powers muscles like the sternocleidomastoid and trapezius. These muscles allow head rotation and shoulder elevation. Proper coordination enables stable posture and movement. Cranial nerve the 12th, the hypoglossal nerve, controls tongue movements important for speech and swallowing. The tongue must move precisely to shape sounds and manipulate food. Hypoglossal function ensures coordinated, fine motor control. Even small disruptions can affect speech clarity. The first two cranial nerves, olfactory and optic, arise from the cerebrum, while many others emerge from the brainstem. This difference reflects developmental and anatomical organization. The cerebrum handles higher processing of smell and vision. The brainstem houses many core motor and sensory nuclei for other cranial nerves. Some cranial nerves are purely sensory, some purely motor, and some mixed, reflecting diverse roles. Pure sensory nerves transmit information inward without driving muscles. Pure motor nerves primarily control muscles. Mixed nerves combine both roles in one pathway. The human body has 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. These nerves connect the spinal cord to the rest of the body. Each pair exits at a specific spinal level. This segmental organization supports structured distribution of sensation and movement. Spinal nerves are mixed nerves, typically carrying both sensory and motor fibers. Sensory fibers bring information to the spinal cord. Motor fibers send commands outward to muscles. This combination allows two-way communication through each nerve. Each spinal nerve forms from the union of dorsal sensory roots and ventral motor roots. The dorsal root carries incoming sensory information. The ventral root carries outgoing motor signals. Their union creates a single mixed spinal nerve. The spinal cord is described as segmentally organized, with 31 segments corresponding to the exiting spinal nerve pairs. Each segment connects to a particular region of the body. This structural pattern helps clinicians match symptoms to specific spinal levels. Segmental organization supports systematic mapping. There are 8 pairs of cervical spinal nerves, even though there are only 7 cervical vertebrae, because of how the roots exit near the skull and upper spine. The first cervical nerve exits above the first vertebra. This creates an extra cervical nerve relative to vertebrae count. Understanding this prevents confusion when interpreting imaging or symptoms. The C8 spinal nerve root exits between the C7 and T1 vertebrae, reflecting the extra cervical nerve pair. This unique numbering pattern continues down the spine. Below that level, nerves exit beneath their corresponding vertebrae. These details matter when localizing nerve compression. Dermatomes are skin areas primarily supplied by sensory fibers from specific spinal nerve roots. Each dermatome corresponds roughly to a spinal level. When sensation changes in a certain skin pattern, clinicians can use dermatomes to estimate which nerve root is involved. Though overlapping exists, the pattern is often informative. Some descriptions note 31 pairs of spinal nerves, but about 30 dermatomes because the C1 nerve typically does not contribute to cutaneous sensation. The first cervical nerve often lacks a typical sensory skin distribution. This explains why dermatome maps do not always show a C1 region. It highlights that anatomical numbering and sensory mapping are related but not identical. The spinal nerve's regional grouping supports structured innervation patterns across the trunk and limbs. Cervical nerves largely serve the neck and upper limbs. Thoracic nerves serve much of the trunk. Lumbar and sacral nerves contribute to lower limb innervation. This arrangement reflects developmental segmentation. Spinal nerves connect the spinal cord to specific regions of the body, distributing sensory and motor innervations segmentally. This means each region of the body has a predictable neural connection. Damage at a particular spinal level affects corresponding areas. This systematic design supports diagnostic reasoning. Spinal nerves carry information to and from the trunk and limbs and are central to reflexes and voluntary movement coordination. Reflexes, such as withdrawal from pain, can occur rapidly through spinal circuits. Voluntary movements also pass through spinal pathways. The spinal cord serves as both conduit and processing center. The spinal cord's segmental organization supports clinical mapping of symptoms to spinal levels via dermatomes and myotomes. Myotomes refer to muscle groups supplied by specific nerve roots. Combining dermatome and myotome patterns allows more precise localization. This method is widely used in neurological examination. Cranial nerves collectively support many head and neck functions, including sensory input, motor control, and autonomic regulation. They enable facial expression, swallowing, eye movement, hearing, balance, and more. Many protective reflexes rely on them. Their combined function allows coordinated head and facial behavior. Cranial nerves coordinate key protective reflexes, such as blinking and swallowing by linking sensation to motor output. For example, irritation of the eye triggers blinking. Sensation in the throat can trigger swallowing or gag reflexes. These reflexes protect delicate tissues from harm. Many cranial nerve disorders are evaluated through neurological examination and imaging to localize dysfunction. Specific tests like checking eye movements or facial symmetry help identify which nerve is affected. Imaging may confirm structural causes. This structured approach reflects the predictable anatomy of cranial nerves. Spinal nerve disorders may be evaluated with physical examination, imaging, and nerve conduction testing depending on symptoms. Weakness, numbness, or pain patterns guide initial assessment. Imaging can reveal compression or structural change. Electrodiagnostic studies can assess nerve function. The peripheral nervous system includes cranial nerves and spinal nerves, linking the central nervous system to sensory receptors, muscles, and organs. It acts as the communication network that connects brain and spinal cord to the body. Without it, central processing would have no input or output. Its reach extends to skin, joints, glands, and viscera. The nervous system uses cranial and spinal nerves as primary conduits, linking the central nervous system to sensory organs, skin, muscles, and viscera. These nerves carry electrical signals across long distances. They distribute commands and collect information continuously. This communication network underlies perception and movement. The nervous system's organization allows rapid reflex-like responses and slower integrative responses, depending on pathway length and synaptic processing. Some actions are almost immediate, like withdrawing from heat. Others involve complex processing, such as planning a movement. The architecture supports both speed and depth. Many involuntary organ functions are regulated through autonomic pathways that do not require cortical involvement for moment-to-moment control. Heart rate, digestion, and gland secretion often proceed without conscious thought. Autonomic circuits handle these tasks automatically. This frees higher brain regions for other functions. Autonomic dysfunction can affect multiple systems simultaneously because autonomic fibers innervate heart, vessels, lungs, gut, and glands. When regulation is disrupted, effects may appear in diverse organs. Symptoms might include changes in heart rate, digestion, or temperature control. This reflects the wide distribution of autonomic pathways. The autonomic nervous system integrates three divisions: sympathetic, parasympathetic, and enteric-to coordinate involuntary physiology across organs and the gut. Each division has distinct but overlapping roles. Their combined activity maintains internal balance. This integration supports stability in changing environments. The autonomic nervous system's core role is maintaining internal homeostasis while adapting to changing demands and stresses. It constantly adjusts heart rate, blood pressure, and digestive activity. These adjustments happen seamlessly in the background. The result is a stable internal environment. Sympathetic and parasympathetic outputs are not simply on or off, they vary by organ and context to fine-tune physiology. In one organ, sympathetic influence may increase activity, while in another it may decrease it. The balance shifts moment to moment. This fine-tuning prevents oversimplified extremes. Across the nervous system, coordinated electrical conduction, chemical neurotransmission, and glial maintenance together enable perception, movement, autonomic control, and internal stability. Electrical signals carry information quickly along axons. Chemical signals transfer information between cells. Glial cells maintain the environment that allows both systems to function smoothly. Tight junction proteins such as claudins and occludin are widely discussed as key components of the sealing between blood-brain barrier endothelial cells. These proteins form part of a molecular zipper that closes the spaces where fluid might otherwise slip between cells. By controlling how tightly neighboring cells connect, they help determine how selective the barrier can be. This microscopic architecture supports the brain's need for an unusually stable chemical environment. Blood-brain barrier tight junctions are not static, and they can change under physiological or pathological conditions. The barrier can adjust its properties in response to signals from nearby cells and shifts in body state. Under stress, inflammation, or injury, the tightness of the barrier may be altered in ways that change what can pass through. This flexibility shows that the barrier is an actively regulated system rather than a fixed wall. Blood-brain barrier studies often focus on both physical barrier properties, like tight junctions, and transport processes, like channels and carriers, alongside low levels of transitosis. In many tissues, cells move substances across by packaging them into vesicles, but brain capillaries are known for keeping that vesicle traffic unusually low. At the same time, the barrier still must deliver essential nutrients through carefully controlled transporters. This combination of strong physical sealing and selective transport helps the brain remain protected while still being supplied. Blood-brain barrier research emphasizes that the very low rate of transitosis in brain endothelial cells distinguishes them from many other tissues. Transitosis is a way of moving material across cells using vesicles, and many organs use it freely. In the brain, limiting this pathway reduces the chance that large or unwanted molecules drift into sensitive neural tissue. This is one of the less visible but highly important features of barrier selectivity. Astrocytes are described as playing pivotal roles in blood-brain barrier formation and maintenance through interactions with endothelial cells. They sit close to the vessels and can release signals that encourage barrier-like behavior. During development and in ongoing maintenance, these signals help keep endothelial cells specialized for brain tissue. This partnership shows how support cells can shape the behavior of blood vessels at a molecular level. Research has provided evidence that astrocytes can induce blood-brain barrier-like properties in non-neural endothelial cells in vivo. This suggests that astrocytes do more than support the barrier. They can actively instruct cells to adopt barrier characteristics. In practical terms, it means the surrounding neural environment influences what blood vessels become. This kind of cellular teaching is part of how specialized tissues are built and maintained. The blood-brain barrier's selective transport means the brain's extracellular environment can remain stable even when blood composition fluctuates. After meals, exercise, or stress, blood chemistry shifts in normal ways, yet brain tissue needs continuity to function smoothly. By controlling entry and exit of ions and molecules, the barrier reduces sudden swings. This stability supports reliable signaling and protects delicate neural circuits from abrupt changes. Blood-brain barrier-associated transport proteins selectively move nutrients and help regulate ionic composition important for neuronal signaling. Neurons depend on precise concentrations of ions like sodium, potassium, and chloride to maintain electrical readiness. Transport systems of the barrier help sustain those conditions by controlling movement between blood and brain. In this way, the barrier is not just a gatekeeper but also a manager of the brain's electrical landscape. The blood-brain barrier's selective permeability is one reason many circulating pathogens and immune factors have limited direct access to brain tissue. In the bloodstream, immune cells and inflammatory molecules can rise and fall, especially during infection. The barrier reduces how directly those changes affect the brain. This separation helps protect neural tissue from immune activity that might be helpful elsewhere but disruptive in the CNS. The blood-brain barrier also constrains entry of many antibodies, which can complicate immune-based therapies targeting the central nervous system. Antibodies are large proteins designed to bind targets strongly, but that size makes them hard to deliver across a tight barrier. Even when an antibody therapy works well in other tissues, reaching the brain can be difficult. This is one reason researchers explore specialized delivery methods for CNS conditions. Blood-brain barrier integrity is clinically relevant in conditions like stroke and epilepsy, where barrier disruption is studied as part of disease mechanisms. When barrier properties change, substances that are normally kept out may enter brain tissue more easily. That can affect excitability, information, and overall neural stability. Studying barrier integrity helps scientists connect vascular changes to changes in brain function. Blood-brain barrier research examines how barrier disruption can allow atypical entry of solutes and signaling molecules into central nervous system tissue, affecting neural function. When unusual substances cross, they may change how neurons and glia behave. Even small shifts in the chemical environment can influence firing patterns, swelling, and inflammatory responses. This is why barrier changes are often treated as meaningful contributors to symptoms and recovery. Cerebrospinal fluid is clinically sampled by lumbar puncture, typically accessing the subarachnoid space. This procedure collects a small amount of fluid that circulates around the brain and spinal cord. Because the fluid is in close contact with central nervous system spaces, changes in its contents can reflect processes happening in or near the CNS. Clinicians can use it to look for signs of infection, inflammation, or bleeding. Cerebrospinal fluid supports diagnostic medicine because changes in its contents can reflect infection, inflammation, bleeding, or other central nervous system pathology. For example, immune activity can alter cell counts and protein levels, while bleeding can introduce blood breakdown products. These clues help narrow down what may be happening in the CNS. CSF becomes, in a sense, a chemical snapshot of the environment around neural tissue. Cerebrospinal fluid is sometimes described as plasma-like, but with a distinct composition and a lower protein concentration than blood. It shares the idea of being a clear body fluid, yet it is more tightly regulated. Lower protein levels reflect the selective barriers that control what enters CSF spaces. This distinct composition helps maintain a stable setting for the brain and spinal cord. Choroid plexus capillaries are fenestrated, but the epithelial layer controls entry into cerebrospinal fluid via tight junctions and transport. Fenestrated capillaries allow easier exchange at the vessel level, yet the system still maintains selectivity. The choroid plexus epithelium acts as the real gatekeeper into CSF. This layered design allows production and regulation without sacrificing control. The choroid plexus epithelium has tight junctions that help control which substances enter cerebrospinal fluid from blood-derived filtrate. Tight junctions keep the spaces between epithelial cells sealed, guiding materials through regulated routes instead of open leaks. This allows the body to set and maintain the composition of CSF. The result is a fluid environment that supports the needs of neural tissue. Cerebrospinal fluid provides buoyancy that reduces the effective weight of the brain within the skull, decreasing mechanical stress on lower brain regions. Without this buoyant support, the brain would press more heavily against structures below it. CSF helps distribute mechanical forces and reduces strain. It's a quiet physical protection that operates constantly, even during simple movements. Cerebrospinal fluid helps remove metabolic waste from central nervous system spaces, which contribute to chemical stability around neural tissue. Neurons and glia are active cells that produce waste products as they run their biochemical processes. CSF circulation helps move these byproducts away from local spaces. This supports long-term stability in the brain's internal environment. Cerebrospinal fluid can transport neuromodulators through ventricular and subarachnoid spaces, linking fluid dynamics to signaling. Not all signaling is confined to synapses. Some chemical messages can move through fluid spaces. CSF can carry substances that influence how neurons respond, helping coordinate broader state changes. This adds another layer to how chemical communication can operate in the CNS. Cerebrospinal fluid supports waste removal and transport of neuromodulators and neurotransmitters within central nervous system spaces. In addition to cushioning, CSF helps manage what accumulates around neural tissue over time. By circulating and being renewed, it can help maintain a clean and balanced chemical setting. This makes CSF part of both protection and regulation. Cerebrospinal fluid is produced continuously and reabsorbed continuously, so its composition and volume reflect ongoing physiological processes. The CSF system is not a closed pond, it behaves more like a flowing stream. Production and reabsorption allow the body to keep conditions within a controlled range. This dynamic balance helps the CNS remain stable despite constant activity. Cerebrospinal fluid is reabsorbed in part due to pressure differences between CSF spaces and venous sinuses in many descriptions. The flow back toward venous pathways depends on gradients, meaning small differences in pressure guide movement. This helps keep CSF volume from building up excessively. It is one way the body maintains steady pressure around the brain and spinal cord. Arachnoid granulations are described as one-way drainage structures in many accounts, allowing cerebrospinal fluid to return to venous circulation. The one-way nature helps prevent blood from flowing backward into CSF spaces. This supports cleanliness and controlled composition. It also keeps the flow direction steady within the overall CSF cycle. Cerebrospinal fluid can also drain along additional pathways discussed in modern literature, highlighting an evolving understanding of CSF clearance. Researchers have been expanding the map of how fluid leaves CNS spaces. These additional routes suggest that clearance may involve more than a single traditional mechanism. This evolving picture reflects how scientific knowledge deepens as tools for tracing fluid movement improve. The nervous system relies on a wide range of neurotransmitters, including amino acids, monoamines, neuropeptides, and acetylcholine. Each chemical family has different properties, such as speed, duration, and typical roles in circuits. This variety allows signaling to be fast in some situations and slow or modulatory in others. It also supports the many different moods and operating modes of the nervous system. Medical references list key neurotransmitters, including acetylcholine, glutamate, GABA, glycine, dopamine, norepinephrine, and serotonin. These examples help illustrate the range of chemical tools used in neural communication. Some primarily excite circuits, some inhibit, and others broadly modulate how networks respond. Together, they contribute to everything from muscle control to attention, motivation, and emotional tone. Synaptic transmission can be terminated by reuptake into presynaptic terminals or surrounding cells, or by enzymatic degradation. Ending a signal is just as important as starting it, because timing is central to information processing. Reuptake recycles neurotransmitters for future use, while enzymes can break them down into inactive components. These mechanisms help prevent signals from lingering and overlapping. Neurotransmitter receptors are proteins that convert chemical binding into cellular responses, including ion movement or second messenger cascades. When the neurotransmitter binds, the receptor changes shape and triggers a downstream effect. Sometimes that effect is immediate electrical change, and sometimes it's a slower biochemical adjustment. This is how a chemical message becomes a functional change in a cell. Astrocytes help stabilize the chemical environment around synapses by regulating neurotransmitter levels and ions such as potassium. When neurons fire repeatedly, the space outside cells can accumulate neurotransmitter molecules and shifting ion concentrations. Astrocytes help clear and buffer these changes. This supports reliable signaling by preventing the environment from becoming unstable. Astrocytes can help recycle neurotransmitters at synapses, contributing to stable signaling and reduced excitotoxic risk. Recycling means neurotransmitters can be taken up, processed, and returned in forms that neurons can use again. By limiting excessive transmitter buildup, astrocytes help prevent overstimulation. This supports long-term circuit health, especially in regions with high activity. Astrocytes provide metabolic support to neurons, helping meet the energy demands of signaling. Neurons use large amounts of energy to maintain ion gradients and communicate rapidly. Astrocytes can supply nutrients and help manage energy-related resources in local tissue. This supportive role helps neurons sustain their work without exhausting local supplies. The nervous system's signaling is energy-intensive, which is one reason neurons depend on strong metabolic support from surrounding cells. Maintaining resting potentials and firing action potentials requires constant ion pumping. Synaptic transmission also consumes resources through transmitter packaging, release, and recycling. The need for steady energy is a fundamental feature of how fast and precise neural signaling can be. Astrocytes regulate extracellular potassium, helping stabilize neuronal excitability during ongoing activity. When neurons fire, potassium can accumulate outside cells, and that can change how easily neurons fire again. Astrocytes help take up and redistribute potassium. This buffering helps keep neural activity from drifting into overly excitable or unstable patterns. Astrocytes also influence synaptic transmission by taking up neurotransmitters and shaping how long signaling molecules persist. The duration of neurotransmitter presence in the synaptic area affects how strongly and how long the postsynaptic cell is influenced. By clearing transmitters efficiently, astrocytes help keep synaptic timing crisp. This supports precise information processing across networks. Many neurons rely on glial buffering of extracellular ions to prevent excessive excitability during sustained activity. Excitability is influenced by the chemical conditions around neurons, not just inside them. Glial buffering helps keep those conditions within safe ranges. This is one of the ways the nervous system protects itself from instability during high-demand states. Glial regulation of neurotransmitter levels helps prevent prolonged synaptic activation that could disrupt circuit timing. If signals linger too long, circuits can lose the precise rhythm that supports coordinated behavior. Glial uptake and recycling systems help end signals cleanly. This maintains clarity and communication across neural pathways. Astrocytes communicate with each other via signaling mechanisms and can form networks that influence local brain homeostasis and signaling. Rather than acting in isolation, astrocytes can coordinate across regions. Their network behavior can help distribute ions, coordinate metabolic support, and influence how local environments remain balanced. This adds a layer of tissue-wide regulation beyond individual synapses. Astrocytes contribute to neurovascular coupling by interacting with blood vessels and influencing local blood flow regulation. When neural activity increases in a region, that region often needs more oxygen and nutrients. Astrocytes help coordinate signals between active neurons and nearby blood vessels. This supports the matching of blood flow to neural demand. Microglia act as immune-like cells in the central nervous system, involved in scavenging debris and responding to pathogens. They help monitor the tissue environment and can change their behavior when they detect damage or infection-related signals. By clearing debris and dead cells, they support maintenance and cleanup. This immune-like surveillance helps preserve healthy neural tissue. Microglia participate in clearing dead cells and debris, supporting tissue maintenance in the central nervous system. Cleanup is essential because lingering debris can interfere with normal signaling and can promote inflammation. Microglia help remove what no longer belongs, allowing the environment to stay functional. This maintenance role operates quietly in the background of everyday brain life. Many central nervous system glial cells, including astrocytes and microglia, can respond strongly to injury or disease by changing morphology and function. They may enlarge, shift gene expression, and alter how they interact with neurons and blood vessels. These changes can be protective, aiming to stabilize tissue and limit damage. At the same time, the balance of these responses can influence recovery outcomes. Astrocytes show heterogeneity and can have beneficial or detrimental roles in blood-brain barrier integrity depending on context and signaling. In some settings, astrocyte signals help strengthen barrier properties and stabilize the vascular interface. In other settings, shifts in signaling may contribute to barrier changes that allow unusual permeability. This context dependence highlights that biology often depends on conditions, timing, and local signaling states. Action potential propagation depends on local circuit properties, including axon diameter and myelination status, which influence speed and reliability. Larger diameter axons generally allow faster electrical conduction because internal resistance is lower. Myelination further boosts speed by reducing current leak and focusing regeneration at nodes. Together, these properties shape how quickly different pathways can transmit information. Saltatory conduction relies on electrical insulation by myelin and periodic regeneration at nodes, allowing rapid long-distance signaling. Myelin reduces the need for continuous membrane depolarization along every segment of the axon. Nodes provide the reset points where the action potential is refreshed. This efficient design supports fast communication across long neural distances. The nervous system's command center role is often summarized as continuously sensing internal and external conditions and coordinating responses through electrical and chemical signaling. Sensory pathways bring in information about the world and the body's internal state. Processing networks interpret that information, and motor or autonomic outputs adjust behavior and physiology. This coordination is what allows a living body to remain stable, responsive, and adaptable. And as you hold that picture signals traveling, barriers filtering, fluids circulating, glial cells steadying the environment, it can be enough to remember that much of your nervous system works quietly without asking for attention, maintaining balance through countless small adjustments, moment after moment,